All right, thank you. So before we get started, I just have a couple questions. First off, raise your hand if you're a chemistry major. Raise your hand if you're not a chemistry major. Okay, all right, that's a good thing to know, okay, for me, so that I don't. I wanna level set this talk right, so that it's not too dry or boring. So yes, last week you all heard Dr. Holmes talk. He was my graduate advisor. So that's what I used to do. I no longer do that. So this talk is going to be pretty different. But I think there's an important message there, though, that if you study chemistry or whatever field you are studying and you really apply yourself to it and learn it well, you can apply it to other things. Because I'm not going to get into too much detail in this talk about it, but the things I learned working on those kind of dry technical problems, it helped me learn to problem solve for a really wide variety of things. So don't hyper specialize, be ready to try new things. And I promise that's the end of like the life coaching stuff because I'm not far enough in my career to be doing much. So let's see. Here we go. Oh, a little bit of a lag here. Okay, so before we go into the talk, I'm gonna give you the outlines, where we're going with things, right? And then you can also get wiggle in your seat if I'm getting close to time and you think I got. So first thing I'll do is a disclaimer. I have to do those in all my talks in industry. Do a little bit more introduction, but then we're going to talk about some questions, right? And we're going to answer those questions. We're going to answer what is chemical characterization within the context of medical devices. We're going to answer why should I care? This is particularly important because all but one of you are not chemists. I'm going to talk about a risk-based approach, managing risk, and kind of a different way of doing it, and something that's applied to a lot of complicated problems. Give some examples in medical device work. I'll talk about kind of what's done in the study, the practical day-to-day operation of doing it, and then close up with some of what the challenges are in the work. And for me, what makes it interesting, right? So as Skipty mentioned, I'm a technical lead, so I'm a team lead senior scientist, and my job within the organization isn't to just do the projects, the routine stuff. I'm kind of thrown at the less routine things. So when you go into work, you'll find that there are two types of people. There's people that love to know what they're going to be doing every day for the next year. There's people that sounds horrible, and to me, that's horrible. So I like taking on odd jobs and problems and things constantly changing. So I'll tell you a little bit of some of the craziness around that, and yeah, and then we'll wrap it up with conclusions and, of course, any questions that anyone will have. So, disclaimer: the opinions, viewpoints here are mine alone. This isn't me speaking on behalf of Eurofins EAG in terms of policy interpretation or current thinking on any of the project stuff. All right, so mentioned this already a few times. Team lead, senior scientist. The things I work on: chemical characterization of medical devices. I also do some particle analysis. This is related to medical device stuff, but it's not all that. So anytime you have a manufacturing process that's pretty well regulated, you're going to be concerned about contaminants getting into your process. And when they do get in, you want to know what they are so that you can try and stop that from happening in the future. You also want to know, does it jeopardize the stuff that you've made? So that's a lot of what particle analysis is. People will take a little jar, throw some particles in it, they'll mail it to us and say, solve our problem, figure this out. What is this? And so we have a tiny sample, right? These particles can be a millimeter across. We can only have 2 or 3 of them, maybe just one sometimes. So we have to work with very limited amount of sample to get as much information as possible, and then figure out what the preponderance of evidence suggests about where this particle came from, what it is, what they can do about it, and whether or not it's good for them. Also surface chemistry. So this gets into like imaging type stuff like SEM, DEM. And then overall, this is process development optimization. So a lot of that. Talk about it later, but the regulations around medical devices are changing a lot. And so the interpretation is changing as well. And so you have to keep up with it. It's not like this is the goalpost. If you make it, you're good. It changes. So we've had projects that were submitted a year previous, and the client comes back and says, the FDA is asking us all these questions. What's going on? And we have to explain, we did finish this project a year ago and you sat on it, didn't turn it in. And so the FDA has moved on. Their thinking on this problem has changed. And so they want to see a little bit more. Of course, we help them work through that, and we're pretty successful at it, but just goes to show that, it does change. So on the right, you'll see a chemical structure, not a quiz. And on the bottom, you'll see a plot of collision voltage, or relative abundance versus collision voltage. And so this is the only point of contact between my graduate work and what I do now. So one of the things I did was those compounds that Dr. Holmes was talking about last week. That was interesting. He may have said molecular self-assembly during the talk, probably. Yeah. So everyone says that in the literature and they just wave their hands. molecular self-assembly. But like no one knows what's actually going on. So I said, how can we find out what's going on? And so what I did, and this was very preliminary work, I don't know that we ever carried it any further, but I took it and we introduced it into a mass spec. And so we could isolate this individual little cluster of, shown up above this iron cobalt square. And then we could collide it with gas and break it into pieces. And we could look at the energies that it took to break the different parts off. And so in this way, we could kind of like probe and interrogate, okay, what are the relative bond energies in this final product? And how does that play into molecular self-assembly? But now I do a lot of mass spec. So moving on, what is chemical characterization? So when we characterize something, it's providing chemical information, right? And it can be lots of different things. But what we're doing is we're doing it for the purpose of informing medical device safety. The standard we do this under is the International Organization for Standardization 10993, Part 18. This is part of a broader section. It's just about biocompatibility. Biocompatibility is this material compatible with living cells, right? So none of you may think of this much because I don't want to get in the room, but most of the things you interact with in your everyday life, there's two options, right? It can be no problem, right? Or it can be a big problem. So your clothing, a lot of the devices you interact with, all that kind of stuff, those go through a lot of extensive testing to make sure that they're biocompatible, meaning they're not going to cause a problem for living tissue, right? So, some of you maybe have heard about nickel allergies with jewelry. That's a really common example of that. So that's an allergic contact dermatitis reaction. And so we're kind of evaluating those sorts of responses as well as many others. There are things that could cause them. And we're doing that pretty much the highest level that we can because we're looking at medical devices. Part 18 focuses on extractables and leachables. So this is not about if I take the whole device and put it on something or someone, is it going to be a problem? It's looking at what leaches or extracts out of it, right? So in common English, extractables and leachables are synonyms, but in this field, they're technically different. So a leachable, you see it's a smaller box down on the bottom right there. So leachables are things that are going to come out of a device under ordinary conditions. That is the conditions of use, right? So if we think about an implant, let's just pick that, right? It's going to be in contact with blood. It's going to be in contact with tissue. It's wet. So it's kind of like saline or basically water, right? Pretty polar. And temperature wise, it's going to be body temperature. Right? Shouldn't really be much higher. If it is, we got a problem, right? So that's leachables. And we can do simulated use studies. And so we extract our device under those conditions, right? We'll do it in saline and we'll do it at 37 degrees Celsius. We'll look and we'll see what we see in it. If there is, what it is, that kind of stuff, right? This information down below. But we want a little bigger margin of safety. So we do extractions. Extractions are heavier. So what we do is we'll elevate the temperature, we'll agitate it while it's extracting, and we'll use a variety of solvents that are more aggressive, right? So we'll do water, a midpolar, nonpolar. Talk a little bit more about that in a second. But it generates these types of information once we analyze it by instruments: quantity, identity, and confidence in that identification. Then from that, we're taking that, we're giving it to a toxicologist. They review it. And that's what's the risk-based approach part. Standard is not the same for every single medical device across the board because it can't be. It doesn't really make sense. So we'll talk about that after we go through motivation. So why should I care about doing chemical characterization instead of the more traditional methods? Well, medical devices are high risk. They can be in prolonged contact with the human body, right? In principle, if you get an implant, it's in you for the rest of your life. So if there's something in there at relatively small amounts that leaches out slowly, but it's a potent genotoxin, that's a problem, right? So we've really got to look pretty hard and pretty low. It can also provide for safer devices because chemical data is unique, right? There are other ways of assessing this. There's traditional methods, right? You'll hear people talk about in vivo and in vitro. So in vivo is in animal models, right? So we take an animal, testing an implant, we plant it in the animal, watch and see what happens. We write it down. That's the study, right? In vitro, so we can have a cell line that we can grow independently and then expose it to the device or to the extracts, much the same way, observe it and see. But what's important is those two methods don't tell us anything chemical, right? If something bad happens to the animal, we have no idea why. We just know it's bad and we shouldn't put it in a person. And that makes it really hard to fix the problem, right? You think about the manufacturing of a device and you start not just the device itself, but all of its packaging and processing materials, all that starting from raw materials found in the environment somewhere, right? and they all go through this manufacturing process. They're cleaned up, they're made into new things, all this stuff, right? And then it's sold to the medical device manufacturer, and they put it all together in this device. You can imagine if there's an entry point for something that's not good, it could be anywhere, right? But if we have chemical data, we can track it down much more quickly, right? And I think the most important one is it's the reduction in animal testing, right? I personally think that's important. Um, some people feel very strongly, some people don't feel very strongly at all, but I think we can all agree that, right, it's probably not the best way to do it, for the reasons I've given you. The other part is that it's not always the most sensitive, right? If we're looking for long-term effects from a permanent implant, how long can we use an animal model, right? Guinea pig only lives a few years, right? So it's good. We can step away from that. And honestly, that's a lot of what the regulators are looking at trying to do with pushing chemical testing over other methods. They want to get away from animals. They want to eliminate it if possible, right? So, risk-based approach. The big question here, should these be treated the same for device testing and approval? So, I apologize. I'm going to ask for a little bit of audience participation. So, I'm just going to ask if you think they should be, well, I'll describe them first. We've got cotton balls on the left. These are being used, jab and alcohol, wipe on your arm before you get an injection. On the right, we have a cochlear implant, permanent. Okay. And my question to you, should these, and I'll tell you all three options so you can decide which one before you raise your hand. Should they be treated the same for testing and approval by a regulatory agency? Your options are yes, no, and I think I want to know. So let's start with yes, they should be treated the same. All right, good. You read the room. I don't think that's true. All right, raise your hand if you think no, they shouldn't be treated the same. Okay, and raise your hand if you think you need more information to make a decision. Good. Yeah. 2 and 3 are both good options, right? 2 is true. 3 is true because that's the approach we're using here. The ISO standard says we need information that is necessary and sufficient to demonstrate safety, right? And the information that's needed is not going to be the same between these two, right? That cotton swab, transient contact, it's a natural product. People have been using it for thousands of years. We know the safety profile of this stuff, right? That's not a problem. But cochlear implant being implanted, it's directly contacting blood and tissue. It's going to be there a very long time. It's very different. The other thing to consider too is bluntly, it's cost, right? If you're making cotton balls, how much can you sell a cotton ball for? Compare that to the price of a cochlear implant. How much testing can you afford, right? And this is something the FDA has to struggle with, right? They know if they demand excessive testing, they will price devices off the market, and that's not good. And I'm not a medical device manufacturer. I don't work for one. I work for contract research organizations. So like I have no horse in this race. It's just that you have to consider if you price a device out of the market, it's not that everyone's safe. It's now we just don't have a treatment option for something. And that's the other part of this risk-based assessment. So this next one, I'm going to describe a situation, and then I'll give you 2 device options, and we'll just kind of see how this goes. So I'm going to tell you about this device. We have some animal model data, and we see that there's some tumor formation after extended exposure. We do a bunch of work looking into it, looking at all details, look at the literature, talk to a bunch of toxicologists, right? Consult the Magic 8 Ball. We expect a few deaths per 10,000 after a decade of exposure. Okay. Is this okay? So I asked the same question. Yes, no, not sure. Yes, no, not okay. Okay, not sure. All right, good deal. Yeah, yeah, I like this. Very skeptical. That's good. This is scientific method in play here. All right, so first device, braces. Well, majority of patients are teenagers, right? So between 10 and 20, you're wearing braces, right? And then you're going to be alive for decades. And what is it to correct? Most times when you're wearing braces, it's not because you're going to die because of your crooked teeth, right? It's just because they're crooked and we want to clean it up. So if this is the case, and there's a ton of people using them, do you think we should approve this based on this animal data? Probably not. I would be impressed. If someone did, I would just have to respect the fact that they were willing to hold that opinion, right? No, I agree with you completely. Then this is completely unacceptable. All right, let's talk about a different device. So this is an aortic stent graft. All right, so how many of you know what an aorta is? All right, good. Everyone's like halfway raising their hands, so I'll say it anyway. So the aorta is this gigantic artery that comes directly out of your heart. It's the largest diameter one, runs down through your middle, spreads out, branches. And so it's, nice arterial blood, it's great for circulation. The only problem is you can have trouble with it as you get older and have certain other risk factors. And so what'll happen is you can get an aneurysm in it, so it enlarges, and eventually it can tear. All right. So if you have an aorta tear, upper aorta, and it's acute, like just like pops open, 40% chance that you're just going to bleed out instantly. They won't even know what it was until they do the autopsy, right? If you make it to the hospital, that 60% chance of survival, the estimate is your chance of survival drops off 1 to 2% every hour. Now, keep in mind, showing up at the hospital and you're like, I don't feel good. Something's wrong, right? We've all been to the ER before and we've seen how concerned they are when you say, I really don't feel good, right? They're like, go over there and fill out some paperwork. We'll be back in 8 hours, right? So 8 hours, that's 16% right there, right? Another thing, above a certain degree of aneurysm, the tear is nearly inevitable. It's basically going to happen. It's just a question of when. And the patients are mostly 65 and older. So with all that information, right, fact that it can be certain that it'll happen under certain circumstances, 40% of the time you bleed out immediately, and it happens in, largely an older population. If, and there's no device out there that's like this is just a thought experiment. If there were a few deaths per 10,000 after a decade, would we be okay with that? I know it's it's a difficult thing to balance, right? It kind of almost turns into the trolley problem, but me personally, yeah, I would take this over that, right? And that's including also the risk of just having the surgery to put it in, a whole separate thing. But that's still better, right? The idea of just walking around with a grenade in your aorta waiting, right? It's pretty terrible. So that's risk-based approaches, right? Though it's not, we're not just drawing a line saying everything has to be above below this. It's we need to look at the full situation and see what are our options and what's the best of the bad choices, right? It's a fallen world. We got to pick what's going to be best, not the perfect thing. So, talked about the why, and I terrorized you a little bit with some thought experiments. Now let's talk about what's actually going on in the lab. So, at the top, I've got this little flow chart. So, when we get a device, we need to test it. There's some calculations stuff we do, but we won't worry about that. What we're going to do first is we're going to extract these devices. So, we're going to put them into containers. We're going to add solvent, shake them, heat them, and just extract out as many things as we possibly can, right? Really over challenge the device to try and make it as safe as possible. Then we're going to take those extracts, we're going to analyze them by a variety of analytical chemistry methods. Then we're going to interpret all that data, and then it's all got to come back together into a chemical characterization report. And in that chemical characterization report, we're going to give analyte information. So it's going to be the estimated concentration, the ID, and the confidence level of the identification. It does require interpretation. There's no system out there where you can just like drop medical devices in and the answer comes out. That's what our clients think we do, but that's not what we do. Okay, we pay a lot of very educated, very intelligent people to sit there and really scrape through this data and take squiggles from different instruments and put it together into a coherent story about this device. It's very rewarding, but it's work for sure. So we do all that, and then we write this report. And I just said I had a bunch of chemists, but we're giving it to a toxicologist. Now they're very educated, but it's a very different type of education. They do know some chemistry, but they don't know nearly as much as we do. Just like we basically know toxicology, except what we hear in conferences. So we have to be careful when we write that report. I'm not writing the report to look cool or get the page count up. I'm writing the report to make sure the toxicologist has only the information that he or she needs to do their job and nothing to distract them or confuse the issue. Right? It's a very different type of technical writing. So extraction conditions. So medical devices can be all sorts of odd shapes. So along the bottom, I've got a few different kind of fake examples. So surgical instruments, for example, can be long and narrow. So we have to come up with ways to do this where we use the minimum amount of solvent. We can have oddly shaped devices. So we can do things like put them in a jar and put beads in to displace volume, or we can seal it into a bag and do it that way for odd shaped things. What's important though is we're trying to extract the device, right? But I just told you we made this system where we're gonna heat it and shake it. And we're gonna use really strong solvents. We got to be picky about our container, right? Because what I don't want to do is do the study, get to the end, and I see more stuff coming out of my container than I did out of my device. And it's like, okay, that makes sense. But then think about it. Like if I challenged any of you, go out and buy something that's cleaner than a medical device that's built for implant. It's like, oh man, that's rough, right? This is not a Home Depot problem, right? This is a little bit trickier. So surprising amount of thought goes into this part, right? We do little studies to see what materials are okay and what kind of problems it could create. Now, if we do see something, we can still address it, but it's not as good as just making sure it never showed up there in the first place. So the other thing we do is we cover the range of solubilities. So like dissolves like, and we want to see everything that's coming out of the device. So we use a polar, a non-polar, and a mid-polar. Polar, you pretty much have one option. You have water, right? And for medical device, it better be able to survive water, right? Otherwise, we're gonna have a problem. Mid-polar, isopropanol, ethanol are two choices. There's some more esoteric ones, but those are the pretty common ones. They're really good at extracting all sorts of stuff. So permanent marker is a great example of that, right? If you write on something with permanent marker and you try and wash it off with water and nothing happens, put a little IPA on it, it comes right off, right? And that's why we use it. If there's something like that soluble in those sorts of solvents, we want it to come off right away. Non-polar, basically we use a variety of like gasolines effectively, right? And this is really good at pulling out non-polar things, and that's important because those things won't go into water at all. So this is kind of a little graph. It just kind of demonstrates that. So on the left, I've got extractables that we've seen in devices, and some of them we see all the time because they're commercially relevant. They're used a lot in packaging. So even if your device isn't made with this thing, it could have it because of the packaging. So um, just four of those. So I've got sodium chloride, a phthalate, furucamide, and I have a colorant, copper 2-thiocyanine. So gloves, that weird blue color that all the gloves have, thiocyanine. Yeah, probably the more common colorant for that, especially the non-sterile ones. So, anyways, we've got our non-polar, mid-polar, and polar, and then I've just marked what the solubility is going to be for each of these things. So, salt, sodium chloride, is not going to dissolve in non-polar, marginally in mid-polar, not a lot, but it definitely dissolve great in water. So we'll get a ton of water, nothing in the others. Phthalates, we see really well in the mid-polars, so we make sure we get a ton out. Lesser in the non and the polar. Uricamide, mid-polar too. The thiocyanine, actually, it's neutral. So it ends up that it comes out in the non-polar really well. And why that's important is when we tell the toxicologist, we don't say, well, in water, thing. No, we report what's the worst case, right? Because we want to be conservative. Worst case scenario, this much comes out in this solvent, right? And they use that, they put that together to do their risk assessment. So I'm going to talk about chromatography, mass spectrometry. It's going to be very brief. If you want to know more, take a chemistry class. They're great. So chromatography is a way to separate out compounds, right? And so what we have is we just very generic. We have a column of some sort, something flowing through it. It might be gas, might be a liquid. We inject on one end, and as the stuff flows through, it flows at different rates for a variety of reasons that I'm not going to get into. So we separate it out, and then if we have a detector at the end, we can see these individual packets of molecules that are all the same within a packet hitting the detector. And the detector will give us some sort of response, usually an electrical signal at the most basic level. It's of course converted into other things so we can understand what we're looking at. But it gives us this little plot, right? And so each of these peaks indicates something that was in that device that came out. And we're able to separate it out. Because if we did that injection at the top, we would get one big blob. It wouldn't tell us there's 3 different compounds. And if we use the right detector, it'll tell us what those compounds are. And that's where mass spectrometry is so useful. So this is really is kind of cheat codes for this type of work, and a lot of others. So the few chemists in the area, it's a huge field and has been for a long time. They're used all over in industry as well as research. So in some way in mass spec, once it comes off that column, you're getting it into the gas phase. Variety of ways to do this. We're not going to worry about it. You also need to create ions. So that process may be just getting into gas phase. If it's already an ion, you might have to hit it with electric or something, right? Variety of ways to do this, but you do that. And then once you have these little bits and pieces of these molecules, they're charged. Anything charged going through an electromagnetic field can be shifted, directed. And that direction is going to be based on the mass of it, the mass to charge ratio. And so what we can do is we can sort out all these fragments, figure out how much they weigh through some variety of detector. There's a bunch there as well. We won't worry about that. But this provides us structural information. That helps us answer that question. What is it, right? The initial signal on the last one from the chromatography, how much? This is what is it? So we don't analyze by one technique. We do a bunch of techniques, and that's to make sure we hit everything. So we'll go through these bit by bit, but basically we do what's called non-volatile residue analysis. We just dry the whole sample and weigh it. This gives us an upper limit. How much stuff is actually coming off of this thing, right? And it's great because mass is the universal detector. It doesn't tell us anything about what it is, but it tells us it's there. Then we've got a couple GC techniques, LC and ICP-MS. So I will go through those bit by bit. Again, it's not going to be a test. The reason we're doing multiple techniques is this. We can plot this big space of mass and volatility. And each technique is better at other parts than others, right? So if something is very volatile, and those tend to be small things, it's going to go with heated headspace GCMS. If it's a little less volatile, but still on the smaller side, GCMS. LCMS comes in as it becomes less and less volatile, and it can go higher in mass. ICP-MS is actually good for elements, so it's basically completely non-volatile, but very low. Non-volatile residue analysis, you can see that takes a huge chunk, but it doesn't tell us any information. And at the top, very large things that are very volatile, it's not really a thing, especially medical devices. You can always come up with some sort of exception, right? Like I'm thinking of, ferrocene, kind of improbable that it would fly, but it does. But you wouldn't see that in a medical device. So we don't have to worry too much about that section. So volatile residue analysis. How much material should we be accounting for by other methods, right? So all we did was we just dry it down and we just weigh it. Seems pretty simple, but the FDA actually puts a ton of scrutiny on this because this is the check, right? So we send them a report. I mean, someone sends them a report. We don't do this. But someone sends them a report and they do this. They report a number and then they sum up everything else they did. And it's like 2 orders of magnitude smaller. It's like 1% that weight. The FDA is going to be like, what are you guys doing? Where did it go? You have to answer the question, right? So very simple technique. We can do it with the balances you guys have here. But it's actually really important because it's that cross check. It's corroborating data. We can also follow up to see what the bulk of the material is because there are times when it's acceptable for it to be less than 1%. Great example of this is if you have a polymeric device, polymer breaks down, but the chunks are still huge. They're not going to fly in an MS. And so you can do some additional techniques to show that it's that. Say, okay, that makes sense. That's allowed. Next one's direct injection. This is semi-volatile things. Use chromatography to separate it. MS for that structural information. We got our identity plus semi-quantitation. This is good for small organic molecules. So polymer's made up of monomers. It's really good at catching residual monomers, right? When you make a polymer, you draw it out in a textbook. It looks real pretty and simple. That's not how it goes at all. It's pretty chaotic. And so what'll happen is you'll have monomers just kind of scattered around or called oligomers. So it's little short chains that are far shorter than what you were trying to do, but it happens. And those extract out really well. And if they're pretty volatile, you'll see them by GCMS. And that's important because some of them can be a problem. Some of you may have heard stuff about acrylates, consumer products. That's kind of a current thing. And that's one example. That's basically what this is. Those acrylates are leftovers from the manufacturing process. They're monomer, and they're not biocompatible. They cause contact dermatitis. So covers monomers, also some plasticizers. You'll see residual solvents as well. Sometimes in a manufacturing process, you might use a solvent to dissolve and recast something, or let's say you're covering something with a coating. That'd be another way you could do it. And if all the solvent doesn't get removed, it'll be there. Those could be a problem, right? All right, LCMS, semi-volatiles to non-volatiles. This is the technique that I started on when I started working at Eurofins EAG mainly. I was doing more pharmaceutical deformulation. Like this is the one I know about. It's a chromatographic method for separation. It gives you MS for structural information. There's some overlap with the GC data. You get the identity plus semi-quant, but because it's liquid chromatography, you don't have to have something that will go into the gas, right? It can still be a liquid. Down the column, you can see a lot more stuff. The instruments themselves too, especially the ones that we have, they're called high resolution instruments. And so they basically give a much more detailed picture, and you can infer a crazy amount of data from a very short data collection time. So very useful. They're good for slip agents, plasticizers, processing aids, surfactants. It's really good at catching. You'll see polymer stuff too, but those are the big things that you'll see there is that kind of stuff leaching out. Again, it's important, right? Everyone here has heard about phthalates, right? ICP-MS is elemental composition. It only runs water samples. Use a plasma jet, and it just destroys everything down to their bare atoms, effectively, right? And this is really good. It's not a chromatographic method, but it's really good at looking for heavy metals. So if those heavy metals are trapped something somewhere, acid treatment followed by the plasma jet, it just blows it all apart. And you can see it. And you can go really low, which is important because things like lead, mercury, and cadmium at very low levels are still a huge issue, right? So we want to know if those are there. So it's kind of an additional thing to address the heavy metals problem. It can also be helpful for other stuff. On LC, the analysts will often look at this data, kind of understand things that they're seeing in the details in their interpretation. So these things aren't completely unrelated. They actually come together in that report. You see most elements, and it's a very precise quantitation, which the other techniques don't necessarily give. Heated headspace. So this one's kind of cool. In the previous liquid chromatography and gas chromatography, to get it into the column, you just take a syringe, you draw up the sample, and you shoot it in. This one has a more elaborate way of introducing the sample. You take the water that it was extracted in, and you're looking for really volatile things. So these are things that evaporate very quickly, very easily, right? Volatile things we're used to are probably smells and fragrances and flavors, right? So what this does, and you can do it for that kind of work too, you have it in the water, you heat the water, then out of the solution, it's into the gas phase. Then you sample the gas phase and see what's in it. And so you can go very, very low in terms of your detection level. And this kind of covers that residual solvents now, gassing issue, stuff you can see. Again, it's chromatographic method, that's structural information, gives an identity, and semi-quantity. What do I do for time? Good. All right. So, yeah, we shoot the stuff in the instrument, generates data. Yay. Well, it's a lot of data. You guys don't know how much data we generate. It's crazy. We still have to have this argument with people in our industry. They don't realize how much an analytical lab turns out. So if you're looking at high resolution mass spectrometry data, on the right, I've got this picture. The top, I've got what's called a chromatogram. So that is what the detector is seeing is all those things are coming off the column, right? So you can see there's hundreds of peaks, right? It's pretty complicated. That's one injection. And for a typical project for LCMS, we'll do over 100 injections, okay? So that chromatogram is really comprised of about 8,000 mass spectra down below. So at each instant in time along that chromatogram, it's collecting a full mass spectrum, right? Boom, 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 boom. This happens 8,000 times, one injection of the 100 that we're doing, right? For this one technique of the 5 that I've described, right? So what that works out to, if I have the data pulled up and I accidentally hit print, it will print a stack of paper 10 stories tall of this from this one technique, right? So the question is, I'm in an industry, so I have to have this done quickly. Can I do that manually in 6 to 8 weeks? There's no way. You can't even pull together a team to do it. And then if you do, number one, who's going to review it? And number two, like what are the results even going to mean? Right? Because if I say, is it right? I mean, I did my best. You told me it was due this day. I got it to you on this day. I don't know what to tell you, right? So we have to depend a lot on tools to automate all the parts that we can possibly automate. That's really important. And I think it's not just about my field. I think it's about a lot of fields where it's like, now we can generate so much data. What's most important is the ability to turn it into something that makes sense, small enough packet of data that humans can understand it and make informed decisions on it. So that's really a lot of what we're doing is helping that process and making sure our automated tools don't go crazy and spew out bad data, right? So it's kind of related to the whole AI thing getting kicked around. Yeah. So putting it all together, NVR tells us how big the puzzle is, right? So we got, some box, we got to account for this much material. What is it all? So we start looking through the different techniques, people working on the different techniques, check each other's work to see what did you find? Do you have more information on this compound? Going back and forth. What's nice is this puzzle, it's not the whole universe of possible compound, right? We're not going to find, compounds isolated from sea urchins in medical devices, right? No one's putting that in there, so we don't have to worry about looking for that. What are we looking for? Materials of construction, a lot of it, right? What is the device made out of? We'll see a ton of stuff from that. Polymeric device, I promise we'll see something that indicates what type of polymer it is. Even though it shouldn't be soluble, we'll see a little bit, right? But then there's also things like processing aids and surfactants, right? So hopefully at the medical device factory, they're cleaning, right? And they do a really good job, but occasionally you will see traces of surfactants, right? And it's fine. They're pretty much everywhere, ubiquitous, but we do see those, right? And FDA okay, you saw it, how much was there, what is it? And what's the risk assessment? Is this going to be a problem or not in a person, right? So most compounds are related to the MOC, and most of them are also related to each other, right? So if I'm looking at polymeric species, if I see a single polymeric species, I'm going to be looking at the other things to see, are these also from this, right? Because the chances of it being one thing, one oligomer from a polymer, and then a million completely unrelated things, very low, very, very low. So you get good at eyeballing it. So that picture on the bottom with the blue peaks, that's a polymer pattern, right? So when someone's reviewing the data initially, they'll open it like, oh, I see a polymer. Yeah. And they may even manually look and just see what the spacing on it is and say, okay, I think it's this and go from there. So most of it is stuff that's fine, right? But the question is, what about the ones that aren't, right? So there may be some degradation. And sometimes you will see things that just don't belong there. And that's why it's tricky, right? You can't fall into the path of making an easy story, saying, well, it's all from the device and everything looks fine. And I had this one compound and it didn't really fit with the device, but I just kind of made it fit with the device. You can't do that, right? You have to look at all the evidence and you have to say, is this thing a problem or not? And we have done it. We have found compounds that were an issue. And that's always fun because you get to tell, so I'm technical. I don't have to talk to the client about it. I tell the project manager and they get to send the email and say, hey, let's hop on a call like now. And then they have to break them the bad news. But it's good, right? We're preventing these things from getting on the market. So what the client sees and what the toxicologist sees is important, right? But turns out it's a lot of really great instrumentation and hardworking chemists in the lab, like so let's talk about challenges. This will kind of be where we wrap things up. There's two challenges to this among many, but these are the ones I picked to kind of talk about. First one is that analytical chemistry is evolving, right? So the techniques for doing this stuff did not just stop developing when we started doing this, right? So they continue to have their development and new instrumental techniques. FDA sees this and they expect people to apply it, right? The other one is that the standards and interpretation are evolving. And I'll get into that. It's amazing how complicated that can be, but we'll talk about that when we get to that slide. So, in terms of chemistry evolving, it's newer, better instruments and newer, better interpretive tools, right? Most of the materials, I won't say all, but most of the materials we're dealing with have been in the market for a while. People understand the chemistry pretty well, which is good, right? Medical device companies, proud to say, are pretty conservative in general, right? They don't like using new things. They want to use things that have a long track record, proven to be safe. It's good, right? So where the big changes come for us, most of the time, it's just better instruments and better interpretive tools. So this graph on the right is it's different. It's mass specs, but it's actually mass specs in space probes. So mass spec instruments get used all over the place because they provide great information. But what I really like about it is we're going from 1971 all the way up to 2023. And basically every single iteration of one of these instruments that was put into space probe was an improvement on the past one, right? So we start with the old quadrupoles and magnetic field instruments. We get into like time of flight and 2 or 3D. So people like basic, almost physics, really designing new detector systems for them, just making them better and better and better, and making resolution better, and all these different performance measurements that they can maximize as best they can. So this is constantly going on. So as soon as you buy an instrument, it starts to go out of date, right? So that's something you have to struggle with. We're lucky we have very new instruments. The one of the ones that we have, so we have a fleet of 4 high-res mass specs. This is me just liking my instruments. We have serial number 5, Thermo Scientific. It's a high-resolution mass spec. The instrument costs $1 million. It's basically hand-built. We had the first one that Thermo didn't install inside one of their own facilities. So we were there at the very beginning, and we've worked very closely with them to lean on them to help us do our job better as well. So kind of an interaction there where we help drive their software development. So, and the last part was big data, right? I talked about that 10-story stack of paper and how we go through that. Well, that's part of that relationship with vendors, manufacturers is the software element, right? And pushing on them to develop better software or take what they have and make it more appropriate for what we're doing. So, for example, compound discovery, the one on the bottom left, that's what we use to process the LC data. And so everyone remembers when there was a chip shortage, right? So we got these instruments in, we were getting compound discovery on computers to run it and stuff. A laptop will die if you try and process one of these data sets, right? 500,000 pages and all that. There's no way. So we're like, okay, we need to get a better computer. So we went to Thermo and said, hey, do you have better computers? They said, yeah, of course we do, of course we do. We're like, how much are they? Like 15,000 a pop. We're like, no, that's not happening. Absolutely not. So I was like, no, we can do better. I know we can do better. And so what I did was I don't build computers, but I know people that build computers. So what we did was we pulled the spec sheet and I was like, look at this. Can you do better? And what would it cost? So some of the people on the team were way into that. They looked at it and they came back and they said, yeah, we can build basically this monster gaming rig and it'll be $3 grand. Like, okay, cool. And they're like, yeah, but so there's a limit. You can only buy one CPU at a time and all this stuff because there's a chip shortage. So we had to take managers that know nothing about computers, like older people, just like imagine helping your parents with Outlook, right? So we're like giving them post-it notes with like this, you need this CPU, go down to Micro Center and buy one. Right. And so they would get their limit one, and then we'd send another manager. We did this. We gradually accumulated all the parts. And then, we built these ridiculous gaming computers. But the you need good airflow for this kind of stuff, right? So the worst part is that the cheapest cases were the ones that also had the like ridiculous LED lights. So we've got this like corner in a cubicle farm where it's like a stack of these things, and they're always like flashing and lighting up. And the first time the CEO came through and saw this, he was like, what is going on? Like he thought we were mining Bitcoin or something. We had no, 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 no. We're working. We're not having fun. I promise you we're working. And we got it all sorted out. But yeah, it's one of those things. Problem solving. Anyways, so that's the challenge from analytical chemistry. We will now talk about standards interpretation of evolving. So I got two boxes here. At the top, I've got regulatory bodies, and at the bottom, I have consensus standard organizations. So a regulatory body is the person that says whether or not you can sell your device, right? These are geographical, right? So in the US, we have the FDA. Japan, you have the Pharmaceutical and Medical Device Administration. In South Korea, you have the Ministry of Food and Drug Safety. In the EU, you have EU MDR, right? Well, what did I say at the beginning? What was the standard that we were using to do this work? It's the ISO standard, right? But they're a consensus standard organization, right? So these four and all the other ones that I didn't bother to list, they can have an opinion on everything in that standard. And it can be that it's bad. And so they can go through line by line and say, we like this, we don't like this, we accept this, we do not, we reject this. It isn't happening to us, right? That kind of thing. And so you've got to know, okay, where's this project going, right? Because EU MDR stuff sails through there if you follow the standard. They're like, we like the standard, just do the standard, we're good. FDA has opinions, and I'm not saying they're bad, but they have opinions, and it's rough. You gotta work with them. You gotta make sure you do it right, because if you try and send an EU MDR report to FDA, they're gonna be like, "Where's this? Where's that? What are you doing?" And at the very least, you have to sit there and go back and forth with them, and they may reject it ultimately. And so all these organizations, they can do that because they're the regulatory bodies. That's their job, right? And it's good. I'm not offended when they push back on it, right? Because ultimately, it's about safety of everyone. And so it's not a problem, but it is nice when it's just like having a class when the expectations are clear at the beginning. It's much easier than afterwards, right? So these guys though, so where do the standards come from? ISO right now, but that's informed by other consensus standard organizations. So the ISO standard took a bunch of ideas from other things that kind of combined them into one, and they're kind of informed by other things. So ASTM is one, AMI's one. So I'm on a technical committee, and that guides ISO or attempts to guide ISO. They tend to do what they want, but it's a committee that kind of guides that, and then USP as well. There's some stuff in there, and so you're probably wondering, I don't understand like what's going on. The teacher example breaks down here because the teacher knows what's going on. They know the subject very well because they're teaching it, right? These regulatory bodies may not know so well. Remember all those things I said about the chemistry evolving? When they start working there, they stop working in the lab. So they're not keeping up with what's the latest, right? Or what's possible or what's not possible. And so it can be a real struggle that way. And so the reason that these consensus standard organizations exist is in a neutral, project-free environment for technical experts in the industry and other stakeholders to interact with each other and provide guidance that then the regulatory bodies can, the ISO can promulgate, and then those regulatory agencies can look at. So it's kind of this safer channel for that, right? Because if I don't have a project on the line, I'm going to be much more frank about what's possible and not possible. And I don't feel like I can't tell the truth because it's but it's like but there's no project on the line. I can't have a client manager. I can't blow my own project, right? We're just talking honestly about chemistry. So this whole interaction here, like there's meetings for these different organizations. So luckily Amy does most things online. I have to travel for that, but like next week I'm going to the FDA because they're having a meeting and we're talking about stuff. So we're gonna go and see what happens. So that's kind of another part of what I do. It's interesting and even further away from, what I learned in grad school. So anyways, that's just, if you're gonna take anything away from this, really complicated. There's a lot of people working on it. It's nice because you don't have to think about it, but you still get the benefit of it. So, conclusions. Uh, Chemical characterization by part 18 has a lot of benefits, right? Gives us information we can't get any other way. It reduces animal testing. It improves the safety of devices, right? Those are all really good things. Um, Chemical characterization is risk-based, and a lot of things are risk-based. As all of you go into different fields, if you do ever get into standards, just about any standard you go into, you're going to find this idea of risk-based assessment, right? And the question is, ultimately, it's like, I have limited resources. How do I distribute them over problems? I don't do it evenly, right? Because some problems aren't a big deal. Some problems are a big deal. I have a brother in the Air Force, and he does risk assessment, but he does it for rockets. And it's weird because it's rockets, completely different, but we deal with the same things every day, right? It's, people being not paying attention to how important something is, right? What's going to blow up the rocket and what's not, what's paperwork, right? So that's risk assessment. You'll see it pretty much everywhere you go if you interact with standards. Um, Chemical characterization is multi-technique and rather complicated. Go too much into that. The best practices in the field are a moving target, right? And that's because of those changes in analytical chemistry and the changes in regulation and interpretation. So I'd like to conclude with that and see if there are any questions.
Does anybody have any questions? Us? What did you do in undergrad that led you to? Perfect. It's
Where do you see the future of your profession going?
That's a good question. So I'll tell a little fun story to break things up and then I'll answer. You ready? So here we go. I'll tell it now because I'm here. And so no one could be worried anymore. But this morning I woke up in San Jose, flying over. Yeah, yeah, oh yeah, yeah, yeah. So, well, it was another important thing I had. So, I was at a meeting, and yeah, and actually the plane was delayed. That was the other part. They thought they had a fuel leak, and it was like I was watching, it was like risk-based assessment. I was like, it's not time to call it yet because I didn't want to terrorize it, right? I was like, it's not certain yet, so I'll wait. But I'm sitting there waiting, and so I was thinking about this because that's kind of what I was discussing there as well. I was like, what is this going to look like? Right now, these projects are very customized to each individual device. And I think what question is kind of coming up in the field is, will it continue to be this way, or is it going to move to where standard is a bit more fixed and it becomes more routine testing? I think a lot of people that are not in the lab, they think it could be done that way, and they want it to be done that way because it's easier to control costs when it's that way, right? You can think about it's always going to cost this much, there won't be overruns, that kind of stuff. And I don't know if it's going to be there ever. It could be, but I don't know. It's tricky. So see.
So you mentioned like when you made that gaming rig computer thing to like jerry rig together, is there anything else you've had to like do that way? Or like have you had to like reach out to someone else to maybe borrow something like that?
Yeah, not borrow too much, but so when we were talking about the extraction vessels, that's actually a place where we've improvised a lot of stuff. So on that slide, I'm not going to go back because I can't go directly to it, but there was for long narrow devices, this is kind of something we started doing. Again, you don't want to extract in a giant vessel, right? So if you imagine you're extracting a surgical device, which is like an instrument, like a pencil, actually, right? Long and narrow, you don't want to drop it sideways in a jar, right? And then there's like a liter of solvent and this tiny instrument in it. That's no good. So what we do is we take Teflon tubing, and we actually order it from Granger. So it's almost as bad as Home Depot. We order from Granger, and then we just cut, and we order rods, and so we cut the tube to the length of the device, a little bit extra, and then we cut the plugs, Teflon as well, 'cause Teflon just nothing extracts out of it. And we clean it really, really well, and then we assemble that. And what's nice is it's soft enough that when you compress it on the ends, it'll form a good seal without needing an adhesive or a gasket, which those leach a lot actually. Historically, people have known that's always been an issue. They're called elastomers, but gaskets are made out of, and it's been a problem for a long time. So that's kind of one of those, I wouldn't say jury rig, but it was definitely like something that we developed internally to deal with that problem because there wasn't anything available commercially. And that kind of stuff is strongly encouraged in the field. When I first proposed this, no one was like, "What are you doing? You're goofing around. Get back to work." It was like, "Oh my God, you might have figured something out." And then they start challenging me. Well, like, "Well, how clean is it? How do you know?" That kind of stuff. So yeah, tinkering is encouraged in chemistry for sure.
How does your field apply or like interact with nursing?
Good question. So pretty much everything a nurse is going to be handling in their daily work, it's going to have gone through this process, right? So like all the syringe needles, all that kind of stuff. And I think it's important too to consider because, when we think about exposure with devices, and we think about the patient, but then there's that part of it's the workers that are exposed as well, right? So, a transient exposure with a device, like let's say handling something, like gloves are a great example, right? Like if you go to the hospital, someone with a glove might touch you a few times briefly, right? If you have surgery, okay, there's a little bit more, but imagine you're the person on that's actually doing the work, right? You're wearing those gloves all the time. So it brings up an interesting point, right? And there is testing for that kind of stuff, but it's not necessarily what we do on our side. We're more focused on patient exposure, but it's the same issues, right? Whether it causes the patient a problem or, a nurse or physician, still a problem.
Thank you.
All right. Thanks, everyone.